research. He is also co-director of the Focused Ultrasound Center of Excellence at Sunnybrook Health Sciences Centre. Dr. Heinonen is also professor in the Department of Medical Biophysics and cross-appointed professor in the Institute of Biomaterials and Biomedical Engineering at the University of Toronto. Dr. Heinonen is a pioneer of focused ultrasound and was instrumental in developing the first clinical system for magnetic resonance image-guided focused ultrasound. He has invented and developed methods for brain ablation and blood-brain barrier opening. Dr. Heinonen currently leads a team of more than 50 engineers, scientists, students, and technicians who design and test systems, conduct preclinical studies, and facilitate clinical research at Sunnybrook and other hospitals in Toronto. Our second speaker will be Dr. Isabelle Obara. She holds a Tier 1 Canada Research Chair in Brain Repair and Regeneration, and she is also a senior scientist at the Sunnybrook Research Institute and a professor in pathobiology at the Temerity Faculty of Medicine at the University of Toronto. The Brain Repair Group, led by Dr. Obara, is using MR image-guided focused ultrasound in preclinical models of neurodegenerative disorders, specializing in Alzheimer's disease. And she's aiming to develop therapies that can halt neurodegeneration and promote regeneration. Dr. Nir Lipsman is our final speaker today, and he is a neurosurgeon and scientist and director of the Harquil Center for Neuromodulation. He is also co-director of the Focused Ultrasound Center of Excellence, all at the Sunnybrook Health Sciences Centre. And Dr. Lipsman is also an associate professor in the Department of Surgery at the University of Toronto. He has been an early adopter of focused ultrasound, and he's looking to bring non-invasive solutions to patients as quickly and safely as possible. Dr. Lipsman leads the way by collaborating with experts in a wide range of disciplines, from neurodegenerative disease to psychiatric disorders and oncology. This approach has led Dr. Lipsman and his team to several world-first clinical trials using focused ultrasound, including the first experience of blood-brain barrier opening in Alzheimer's disease, amyotrophic lateral sclerosis, or ALS, and primary and secondary brain tumors. While you are listening to today's talk, feel free to share any questions in the chat, and we'll get to them during our question and answer period at the end. So without further ado, I'd now like to call Dr. Heinonen to begin the first portion of today's talk.
Thank you, Carol, for that introduction. So during the past 30 years or so, there has been a huge development in how we diagnose brain disease. And brain disease, of course, is huge burden for the healthcare system and for many individuals. So if you have a headache and you go to the emergency room, you get an MR scan or CT scan. And if they don't see anything, that's usually good news. But if you get, if there's a tumor, for example, you might get more imaging with PET maybe and seeing what metabolism is doing there. But that's where the good news ends. The therapy is still similar, that a long time ago, requiring cutting the skull and invasively going and removing the tumor. So for many years, I have been thinking, okay, well, the imaging is so great. We can do this non-invasively. We can see what's inside the brain. Why do we need to treat the brain invasively? Can we use this energy that we are using to image also for doing therapy and doing surgery, killing the tumor or delivering drugs? And when you think about the physics of it, obvious thing would be that maybe what can we use? We cannot use light. Light doesn't penetrate deep enough. We don't want to use, we can use radiation, but radiation as ionizing leaves side effects. And we cannot use microwaves. They cannot penetrate deep enough. So can we use something else? We can use ultrasound. Ultrasound penetrates well. And what is ultrasound? Ultrasound is a compressional wave. So if you have a source here, it vibrates back and forth. That pushes molecules and causes a mechanical wave to propagate. That propagates as a tensor compression of molecules or dilation of molecules. And then ultrasound is similar to sound that you hear, except it's a higher frequency. Typically, for therapy, we are talking about 200,000 hertz of vibrations a second, up to 1 million vibrations a second or so. And it's, like I said, it's a mechanical wave and it's non-ionizing. So it doesn't leave any after effects if you are not going over a threshold. And it's, of course, the leading diagnostic method, imaging method in the world. And it's almost, maybe all of you have been imaged before you were born using ultrasound. So it's extremely safe at low exposures. So why are we proposing it for brain therapy? So the ultrasound has this wonderful thing that you can, the wavelengths are so short that you can just curve this source like here, and that means that the waves are propagating to a sharp focus. And if you look at the intensity distribution or the energy distribution along the central line, you can see that it's in the near field and all the energy is concentrated at this sharp focal spot, a few millimeters in diameter. And you could imagine that now we could take this energy and non-invasively put it in the brain and do surgery, for example. So how can we do surgery with ultrasound? So here are now the ultrasound bioeffects plotted as a function of temperature. Here is a temperature in Celsius, and here is the exposure time. And this red line represents the threshold above which you coagulate tissue. So if you turn the ultrasound intensity or the energy high enough, you can elevate the temperature really fast at the focal spot. And if you have 1 second exposure to about 60 degrees, you can coagulate the tissue. So you can non-invasively denature the proteins and coagulate tissue. And of course, you can go higher and you can boil tissue. If you coagulate, you can go from completely coagulated or killed tissue in the brain to completely normal brain within a few cell lines, about 200 micrometers or so. So sub-millimeter going, so very, very high precision surgery. And then, of course, you can drill holes if you boil tissue. And then there is a host of other effects that we talk a little bit about later, which are not temperature related. So ultrasound has these characteristics we can do surgery. So let's talk about surgery a little bit first. So it was proposed a long time ago. 1942 was the first time when focused ultrasound transducer was built, and it was tested to do brain surgery, coagulate brain tissue or cook brain tissue, and they demonstrate it can be done in a preclinical model. But then 10 years or 2 years later, they published another paper which showed some of the problems associated with the focused ultrasound. And the main problem is that, illustrated in this figure, when you have an ultrasound transducer and you're sonicating soft tissue, so launching the wave to soft tissue, and when you hit a bone surface, there is a big problem. Bone is has a higher ultrasound absorption, so energy is lost much faster. And here is now the absorbed energy or power as a function of depth. So you have this exponential decay when you propagate into a tissue, and then you hit the bone surface and you have very high energy absorption, meaning you get a hotspot. So what will happen when you try to go through the skull bone is that you're going to burn the bone and the skin over it, tissues over it. So from that paper of 1944, it had been believed for about 50 years that it's impossible to use ultrasound to do brain surgery to intact skull. So that was then learned by a group in Illinois, Francis and Bill Fry, and they developed a system that had 4 focused ultrasound transduced focused to a spot. But because the skull problem, they did the surgery during in operating theater by removing the skull bone and going to the brain. And they did about 100 patients on Parkinson's. So ultrasound can do neurosurgery, but not through skull. And there are other attempts also, again, removing skull bone and heating the tissue. So how I became interested in focused ultrasound was for brain was this paper. This is Frank Fry and Steve Goss. They published this paper where they used a preclinical model and had a human skull bone, and they sonicated through this skull bone and demonstrated that there was ultrasound energy. They were able to produce a coagulation deep in the brain. About 10 years later, I saw Frank in one of the meetings early morning. He was there, and he was alone in the restaurant, and I went to sit with him, and we had a nice breakfast. That was the only time I saw Frank. It was his end of his career and the beginning of mine. And I asked him about this experiment, and he said that, yeah, it works when the skull is just right. And he kind of gave me doubt that this would ever be able to use in the clinic. But the idea was in the brain. So the idea that I thought that we could do is that we can build a really large transducer and go around, and that way have enough energy to avoid the skull heating, because you could cool the skull and then you distribute the energy and you can do a sharp focal spot, and maybe you can coagulate deep in the brain. And so that was the idea. I built an array. This is 64-element array. This is 30 centimeters in diameter, like a hair dryer, and tested it through the skull. You had human skulls given as a gift by people. And when you put this skull in front of the beam, instead of a sharp focus, you get this kind of blurred focus. And the reason for that is that the skull focus thickness varies and density varies, and it defocuses the focus. And so people think that you couldn't focus through the skull. That was another reason for it. So the solution to that is to tailor each of the signals going from these transducers to correspond to the thickness of the skull bone and the density of how the wave propagates through it. And we did extensive measurements to get the skull properties and being able to have a computer model that will allow us to adjust the signals such that we get a sharp focus. So does it work? Yeah, it works. This is now this focal spot through the skull when it's corrected. And that worked such that it eventually was adopted to a clinical system that is commercialized by Insightec and is now in clinical use. So this is the very first experiment that we did in a human being. And what it showed was that we can get the sharp focus and we can see the focal spot in the MRI scanner. Showed the feasibility. And now there are over 100 hospitals using this device, and over 6,000 patients have been treated mainly for tremor treatments. And Nir will talk more about this. Here is the lesion in the middle of the brain. Here you can see the temperature elevation. So I don't want to talk more about this. Nir will tell you all the details and the fun stuff that he has been doing with it. So the other thing which I'd like to talk a little bit, not too much, is that there is another barrier with the brain, and it's called the blood-brain barrier, where the endothelial cells of the blood vessels are tied so tightly that the blood vessels become like tubes that do not allow very much penetration into the brain. And this prevents most of therapeutic agents to entering in the brain, and has been a big problem for brain therapeutics. And the idea was that, okay, well, now you have this highly focused mechanical energy. Can you just heat it a little bit, this brain tissue, and make the blood-brain barrier leaky, and then use the blood-brain barrier elsewhere in the brain to protect the brain? Because many agents have a positive effect in some location, and then they might be negative effect on another location. So that was the idea originally, and there have been some papers which show that when you produce coagulation, you can have this kind of blood-brain barrier breakage. And we tried it for in preclinical experiments for about 5 years to get the parameters right, and it just didn't work until one day we just, okay, well, let's test these ultrasound contrast agents. So we injected this contrast agent in the bloodstream, and what they are these microscopic gas bubbles, about 2 to 3 micrometers size, and they use as a scatterers for ultrasound, so you can see where the blood vessel is in ultrasound image. So we injected those, and what the important thing with these gas bubbles are that they expand and contract much more than the surrounding tissue. So they are very, very effective energy concentrators. And now when they are in the blood vessel and capillaries, they can expand the blood vessel and stretch these endothelial cells and make the blood-brain barrier leaky. And that can be done at very, very small energy levels. These are like, you need like 10,000 times more energy to create these thermal lesions than you can open the blood-brain barrier. And this is a preclinical model showing a contrast agent coming into the brain, very sharply focused, localizing, and works well. It has been used with siRNAs, viral vectors, nanoparticle cells, and many other things. It looks like everything that we try, we can get to the blood-brain barrier. And it seemed to work in human beings, which was quite a big surprise that it works about the same in humans than preclinical models. And here we'll talk about those clinical trials more, and Isabel will talk about the agent delivery and other preclinical work, how we can use it for brain work. So I'll concentrate a little more on the technical side. So how can we determine when we have the right amount of ultrasound? Because we know that the skull is distorting the beam and so forth. So when these ultrasound, the bubbles are vibrating in ultrasound field, like you can see here, they act as a sound source. So you send an ultrasound beam to the bubbles and they contract and expand. Oops, so go the wrong way. And so they send a sound wave, and when you listen to that sound wave, you can see that we see the fundamental frequency. So you send 500,000 cycles to the bubbles, it scatters 500,000 cycles. So this is the tone, it's very monotonic. And then it scatters also second harmonic frequency, so it has this overtone. And then if we increase the pressure amplitude, the power that we are applying to the transducer, at some point, there is this event that happens where we start seeing these ultraharmonics or subharmonics. So half of the frequency of the ultrasound that we are driving appears scattered from the bubble. And that means that the bubble is starting to have these shape changes, and that is creating the harmonic signal coming out. And that turns out to be a very great internal calibration point. And it turns out that if we look in the histology what's happening in the brain, so if we sonicate at that point and then we drop the energy to 50% and sonicate at that level, we get nice increase of permeability of the blood-brain barrier without any histological effects. But if we keep that at 75% or 100%, we get histological effects and tissue damage. So this allows us now to calibrate the exposures and use them in clinic. So this is, of course, team sports. Here is Isabella and Nir and many others, Megan O'Reilly, who have been doing lots of work for this over the years. So first we have, of course, the physics team and lots of engineering, biophysics. Then we need to have funders to fund the research. And then we had the biology and neuroscience and cancer to look how this technique would work. And then we need to have an industry collaborator who can take and commercialize it, and then clinical. The key piece is, of course, the clinical testing and proving how well it works. And what we have worked with the neurosurgeons mainly, and brain scientists and cancer. So just the last few minutes, what's the future of this technology? So you will hear from Isabel and Nir about some of the near future with the current devices, but the future, you saw the numbers, 1 billion people are suffering brain disease. So using an MRI scanner, we can do maybe thousands of patients a day treatments, but we cannot address all the issues that we need. MRI scanners are not globally everywhere, and there are not enough of them to treat all the patients. So we need to move somehow to tens of millions of treatments a day from the thousands of treatments that is possible with the current systems. And how can we do that? So the first thing is we need to get rid of the MR scanner. We need to be able to do the imaging with the same device that we are treating the ultrasound. Here is an experimental array where we have these ultrasound sources that can also listen the sound. So we transmit and we'll listen. Here is a preclinical model where we were sonically in this grid. And then when we sonic in that location, we captured all the data. And here you can see the image of those bubble vibrations in vivo, in the actual blood vessels and showing where there's. So now we can localize that where things are. To use this to then control the treatments and do it without MRI scanner. The other thing is that can we do it more precisely? Can we actually see the single bubbles? So we have these bubbles floating in the bloodstream. Can we see single bubbles? See 2-3 micrometer bubbles. So we did experiment where we put a tin tube and then used a human skull in front of that array. And then we looked, it did imaging. And here you can see, this is now that spiral. This is 1 millimeter. You can see the single bubbles to human skull with the resolution of better than human hair. So we can control now these bubble vibration with that resolution. So you can see that we can start doing microsurgery or microtherapy in the brain. So that is not in the near future, that is a little further away in the future, but that I think that's possible. So we can see where we can go with this technology. And then the final thing which we have been looking in is to develop a custom arrays for patients where we take the CT scan and then we print a helmet that fits around the head, put transducers on, and here is one helmet testing how well it would work. So that is maybe the future. Maybe each of us will have a helmet at home and we get the weekly or nightly brain cleaning exposure. Who knows? So as a summary, what I have told you is that focus ultrasound can be focused deep in the brain. And it can be guided and controlled by the MRI. It is now a clinical treatment in that regard. It can modulate the blood-brain barrier permeability, and it's also can modulate the brain function. I haven't talked anything about that. There is lots of information on that. So it can also modulate brain function. And the future is it will revolutionize brain treatments and diagnosis. And after you see Isabel's and Nir's talks, I think you will be convinced of that. I'd like to thank many people. This is my group, one of the summers. Lots of bright, brilliant people who are making all this work possible. And then, of course, funders, Western Family Foundation and Sunnybrook Foundation have been key funders at this latest part of our research, and then industry collaborations and many clinicians, just only a few are mentioned here. Thank you so much. Okay.
on one application that Clairvaux mentioned with using microbubbles and blood-brain barrier permeability, and these applications for Alzheimer's disease. We're getting the slide change. Thank you. All right, so first, just to briefly recap what Alzheimer's disease is, it's really like the vast majority of the dementia case are due to Alzheimer's disease. The symptoms include like a deficit in learning, logical thinking and memory, change in mood and personality, problem in recognizing friends, and eventually even family members. And at the end, it's really the inability to communicate, to function, and to live with good quality of life. Sorry, no problem.
Just keep going.
All right. So the prevalence of Alzheimer's disease is about 1 in 10 for people over the age of 65, and it keeps going on as agings progress. So 1 in 4 people over the age of 85, and 1 in 3 seniors will die of dementia. And the risk factors really, the main one is age. The first one is age, and we are all going throughout aging. So I think it's very important that we think about that as we go along in our research to find a way to help Alzheimer's disease. There's family history, head injuries, heart disease, lifestyle. And when it comes to these, really, for heart disease in particular, when you think of heart disease, in general, what is good for the heart is good for the brain. And you can adjust your lifestyle to that. There's no treatment available to stop disease progression to date. And that's what we're working on. In terms of the change in the brains, it goes from microscopic changes that I'll talk about those a little bit through the talk, like those amyloid plaques, the tau tangles, and it affects brain cells, mainly the neurons that tends to degenerate with disease progression. And how does the glial cells react to the pathology as well? When you look at the structural changes, here, if you have like a normal brain on this side, you see my pointer? Yes, that's good. Normal brain on this side. And during the talk, I will focus on the hippocampus, which is a site of memory formation. So it's heavily involved in learning and memory. And you can see with Alzheimer's disease how the hippocampal formation degenerates. There's a lot of other global changes in the brain of an Alzheimer's patient. And we'll talk about some of these changes as we go along. So I will really focus on the drug delivery and what are the challenges that we face. Clairvaux mentioned it, the blood-brain barrier. And this is true for any access to the brain to treat Alzheimer's disease, Parkinson's disease, or brain tumors. So in terms of the drug delivery, I can show here, this is just like blood cells going through the blood vessels of the brain. And of course, it prevents any infection that you may have in the blood to not get into the brain, but it also prevents 100% of the large therapeutic and 98% of the small drugs to actually get to the sites that they will make an effect. So the idea here was like, how can we look at therapeutics that we prepare in preclinical models and make them go through the brain, through the blood-brain barrier using focused ultrasound. So that would be like a non-invasive way to really deliver those therapeutics to the brain. I don't know why the video is starting over again when I click, but bear with me, thank you. So the way that it's working is, it's always fun to use a different computer. Here you have the sound waves like Clairvaux mentioned and the oscillation of those microbubbles. And when those therapeutics here, the small blue points that are injected in the bloodstream, you can see the microbubbles will extend the tight junctions of the blood-brain barrier, and now the therapeutics can go in. I will use the arrow right here, maybe. So this is, with regard to Alzheimer's disease, we know which parts of the brain are really affected. So the beauty of using a guided system here by MRI is that we can actually target really precisely the areas of the brain that are affected in the temporal lobe for the hippocampal formation, the entorhinal cortex, any part that we have the disease pathology being present can be targeted. So in the preclinical research, we also have the ability to control all the variables, or most of them, that we have to really make the experiment and work and test what we want. So the first idea that we had is like, how can we actually study the effect of the microbubbles, the interaction with the ultrasound, and what would it do to the brain, even without the therapeutic? So that was our controlled setup, just microbubbles and blood-brain barrier permeability. And what we found is that actually, if you look here on the untreated side of a preclinical model, the decrease in the plaque load by about 20% with focused ultrasound and microbubbles without any therapeutics into the bloodstream. There was also a change in glial cells. So these glial cells are support cells and they provide nourishment to the neurons. And what we find here on the untreated side, those are the normal morphology of glial cells. And with ultrasound, you can see closer to the epicenter, if you want, of the ultrasound, like the astrocytes, the microglia here are activated, they're slightly larger. And as you move away from the ultrasound spot, they become like more in their normal feature. So I want to point out also that all the work done by the trainees, and I will list them at the bottom here for the data that are present in these publications. And without the undergraduate student, the grad student, the postdoctoral fellow, the research associate, and the technician, this work will not be done. So a big shout out to all of them, because that's how research is done. And now if we look at the mechanism a little bit, what we found is those astrocytes here and microglia, if we just look at all the glial cells, they have the capacity to phagocyte, to basically chew up these amyloid plaques and be able to clear them from the brain. So by being activated, the morphology of the glial cell is changing and it starts getting to the plaque, and we actually found more amyloid deposit within these glial cells. So that was an evidence that they were contributing to the clearance of the plaque. I'm trying to change the slide again, other arrow. Get the chance to see the video again. And then the other discovery that we have with the modulation of the blood-brain barrier is like we actually increase the number of new neurons in the brain. And this is called the process of neurogenesis. If you look at these cells in the brain here, the cells in red are the ones that are dividing. So those are new brain cells that are being born in the hippocampus. So that's the site of memory formation that I told you about. And with the ultrasound treatment, we found an increase in those number of neurons and also their branching complexity. And by branching complexity, I mean these long, they're called axons here, and there's dendrites as well. And that's how the cell communicate from the cell itself to other cells of the brain. So that neurogenesis is increased by 2 to 3-fold with ultrasound and blood-brain barrier permeability. And you can see here, like just representing the cell division and how they start extending their process. And we actually found that these cells mature and integrate well into the hippocampal formation. So to summarize the part of focused ultrasound with blood-brain barrier permeability, really, when we look at the glial cells and the neurons and the way that they respond in a brain that has a lot of pathology, it seems like the effect are pushing like the regeneration mechanism so that we reduce the pathology. We have a mild and transient activation of the glial cells. We found also anti-inflammatory milieu that is being created. It's an inflammation acutely, but with time, it becomes more a pro-regenerative milieu, and it's produced, the glial cell also produce growth factor. And there is neurogenesis and also cell surviving and regenerative signaling going on into the brain. And all this together in our clinical model, we found that it was able to increase learning and memory, so actually get to a functional level. So now in terms of what else can we do? Well, now we know ultrasound alone with microbubbles, blood-brain permeability is having a positive effect. What if we were to add therapeutics? So these therapeutics can be molecules like antibodies, can be vectors for gene therapy. They can also be stem cells. And that work was started over a decade ago. And these were the first papers that we had for each of these different therapeutics. And I'm going to give you some more recent examples now. So this example here is with antibody called IVIG. And those antibodies are taken from thousands of patients, and they're just regular antibody that are in the system of everybody. So they are from the Canadian Blood Bank Services. And thank you to Dr. Branch with that collaboration. So this was 2 treatments of focus ultrasound. The antibodies were given intravenously as well. And there's a lot of data, but I'm going to take you through this. So if you look first at this column here, which is just the antibodies, no ultrasound. And what happens there, we found 0 antibody that would get into the brain. So without ultrasound, the antibody was not detected into the brain. There was no neurogenesis being increased. Inflammation was unchanged. There was a reduction in plaque load. Apologies for that, going too fast. There was a reduction in plaque, and that was based, this antibody, the IVIG, is actually in clinical trial. It was in clinical trial for Alzheimer's disease, and it's been shown to have some efficacy, and it stopped in a phase 3 clinical trial. So it had some efficacy, but not enough to move further. And that was the idea, that we could take this antibody and with ultrasound get more into the brain so that we would increase the efficacy. So we found that it reduced the plaque load as it did in some of the patients for the clinical trial. Now, if you look at ultrasound alone, there was, of course, no antibody getting into the brain. Neurogenesis was increased by 2-fold, like I showed you before, just the effect of ultrasound and microbubbles. There was a reduction in plaque loads. And the really big effect is when we combine the antibody with the ultrasound, where we get a substantial amount of antibody getting in, more neurogenesis, reduced inflammation, and a larger decrease in plaque load. So really the combination of the advantage of IVIG as a therapeutic in the bloodstream and focus ultrasound is making the difference. And the way to think about focus ultrasound is really to think of which therapeutic will be beneficial in the periphery, because they're all injected into the blood, and will it be better once it gets into the brain. So because we had this increase in neurogenesis, and also we see now that the plaque reduction is actually a readout that is quite achievable, we thought, what if we can go further and really boost the capacity to protect other neurons, not only the new neurons that are neurogenesis, but the existing neurons that are into the brain. So this was done in collaboration with Dr. Yuri Saragovi, who produced the molecule D3, which is a growth factor molecule. It actually started at a CAN meeting, so I wanted to mention that. And I'll get you, so here what you can see in red is all the spots that we targeted with focus ultrasound to be able to deliver the therapeutic. Here we just put a dye so that it would be labeled in red, but we targeted the basal forebrain, which is a site that from there, it projects to the entire hippocampal formation and to the entire cortex. So we can get a good coverage of important neuronal pathway involved in learning and memory. So and we did different treatments. So here you can see that this molecule D3 is given into the blood with focus ultrasound. It's been released into the brain. It acts on several receptors, and it can trigger regenerative pathway and pathway that will produce cell death. So we wanted to avoid these pathways and really focus on the pathway that are regenerative and see if the therapeutic can have this effect. So this is many years of work in one table, so I'll lead you through it as well. First of all, like the delivery to the brain was achieved only with focus ultrasound, otherwise the therapeutic does not get in at all. And then we also found that focus ultrasound was letting in some other growth factor, here nerve growth factor. So this was a new finding. And the pro-survival signaling was increased by 2.5 to 3-fold with ultrasound and more with the therapeutic. And the readout that I have here, what I mean by that is like if all the pro-surviving signaling that we looked at with ultrasound only, there was one of these readouts that was increased. So ultrasound would promote one of these readouts. But with the therapeutic, 5 out of 5 readouts that we had will be increased. In terms of protecting existing neurons that we targeted, again, in this case, it's only with ultrasound that we get the therapeutic in that we can protect the existing neurons. So 5 out of 5 readout there. In terms of neurotransmitter release, so this is the chemical that is able to communicate from one cell to the other to be able to function. And it was, again, only with ultrasound and the therapeutic molecule. The production of new neurons also was greatly increased with the molecule and ultrasound, and again, in more readouts. Reduction of plaques, same thing. Therapeutic and the ultrasound is the winner in terms of the strategy. And especially when it comes to function and learning and memory over time. So this is a long time course of experiment with 3 treatments, but 5 of the 5 readouts were increased with the therapeutic and ultrasound. So on that, now you can see from 1 treatment to 3 treatment with therapeutic, there's more benefit. So the vision here is like, what if we could have 1 delivery and a lifetime of clinical benefit? How can we do that? And one way to do this is using gene therapy. So I'll briefly explain what gene therapy is. So if you take a therapeutic protein, for example, an antibody, we know that they're getting at low level into the brain, and we need repeated administration to make these neurons survive. So what if we were taking a recipe for this protein? So this is the gene itself, and then deliver that gene 1 time and use the genetic code to produce these therapeutics over and over again. So really what would happen, the gene will get into the neurons, and the neuron itself would be producing this therapeutics as a mini factory. So it will become its own factory to produce the therapeutic and have positive effect. So the challenge are, you need a therapeutic gene, a gene carrier. I'm taking you to the process, and then you can target with ultrasound the brain, the brainstem, the retina, any part that has a barrier that is difficult to reach from the blood to that site. And then you need to get that gene into the cells that you want, either neurons or glial cells. So as you can tell by the list of trainees that work on that and the publication, it's a huge effort in the lab, and it's a very active component of our project to date. And I will show you just a couple of results here to give you an idea of how it looks like. So if we have, for example, gene carrier 9, here what we're doing is testing a lot of gene carrier to find out which one will be the best, which one can we use. Gene carrier 9, with the gene here in green, you can see the focus ultrasound spots are really clearly labeled. So it means like the gene carrier was able to get into those focus ultrasound spots. This one was not targeted with ultrasound. Here we have the gene carrier that is called PHP.B, and it was not getting in those settings. So this one will not be a good choice for moving forward. And then the next steps, like which gene will be transferred to neurons and glial cells. So we were able to have some gene transfer to neurons. I will show you one example of therapeutic efficacy with gene therapy. And here it was with the gene carrier 9. It's in a preclinical model of Parkinson's disease, where the substantia nigra is an important part that is affected in Parkinson's disease. And you can see in green, the gene was getting in only in the ultrasound targeted area, but without ultrasound, it was not getting in. That therapeutic was made to reduce a pathology related to Parkinson's disease, which is alpha-synuclein. And you can see here, there was, well, if you look at that picture, this one is the one treated with ultrasound and the gene carrier, and there's less of that red signal. And when we quantify it, there was 50% reduction in the alpha-synuclein pathology in this model with the gene therapy. I lost my pointer. So in summary, I think in those preclinical models of neurodegenerative disease, really transcranial ultrasound combined with intravenous microbubbles can reduce pathology, promotes regeneration, and improves cognition. All of these effects we found were augmented with the right therapeutic delivery using focused ultrasound, particularly with repeated treatments. And really the long-term dream a little bit like Clover was mentioning for the ultrasound in terms of the therapy as well could be that for a one-time ultrasound gene delivery, we would have a therapeutic that is produced in the brains for decades and then therefore providing long-lasting benefit. For those of you who are interested in reading more, like this is very recent, it just came out, and it gives also an overview of the work done by many groups in the world on focused ultrasound, both at the preclinical and the clinical level. So it gives a broad coverage of that. And finally, I would like to thank all of the collaborators. There's actually too many to even list. I'm missing some, I'm sure, all the funding agency. Thank you.
Give me 1 sec.
I forgot to introduce me.
Perfect. Okay, well, thank you so much. Do I use the computer to move it, or do I use this to move it? The computer, perfect, okay. Perfect. Okay, well, thank you so much, Dr. Sherman, and thank you so much for joining us. It's an absolute privilege and pleasure to be a part of this panel and follow up with some of the clinical work that we've been doing at Sunnybrook and Sunnybrook Research Institute. My name is Neil Lipsman. I'm a neurosurgeon, and my day job is to open people's heads and treat them that way. So treat brain tumors, hemorrhages, and other kinds of things. And here we are talking about less invasive approaches. So why are we doing that? What is the future of this technology? That's what I hope to cover in the next little bit, but just an absolute privilege to follow the tremendous work that you've seen by Dr. Heinen and Dr. Obara. So just like Dr. Obara mentioned, I often start my talks with this picture just to reinforce just how much work goes into a lot of the work that you'll see. You'll hear from me representing the tip of the iceberg, but just an amazing team that makes it all possible that I'll thank at the end. So before we get to the what we're doing, let's get to the why and why we're doing what we're doing. You've heard a little bit about this before, but brain diseases are complex, of course, but you can summarize them and understand them by 3 words. They're serious, they're common, and they're expensive. So when you look at the top 10 World Health Organization causes of morbidity and mortality, 5 of those are going to be brain diseases. So these are common and they affect basically everybody out there listening and everybody around the world. So they are very expensive as well from a public health expenditure perspective. The global cost of Alzheimer's is estimated to be $1 trillion by 2030. So there's an urgent need to develop safe and effective approaches for these diseases. Proportionate to the clinical need are the challenges that are involved in treating them. So again, a very complex question, but you can summarize them oversimply in 3 words: safety, technology, and access. So it's not enough to have an effective treatment. You want it to be a safe treatment. You want to balance the risks of something with the balance of the benefits of the treatment that you're proposing. There's also been a tremendous gap in the field between our ability to understand illnesses and see them on an MRI, for example, brain tumors and their genetics, et cetera, but not so much in our ability to treat them. So technology has the potential to close that gap between diagnosis and treatment. And finally, you've heard a lot about a physical barrier that exists, the blood-brain barrier, which overcoming that has been a holy grail for a lot of people in the clinical neurosciences for decades. How can we provide a window into the blood-brain barrier, into the BBB? So in these ways, Sunnybrook and Focus Ultrasound has been leading the way. So this is where we work, and this is where we're coming to you from and speaking to you from. And again, the tremendous privilege to work alongside the teams of Dr. Heinen and Obara in what is really the epicenter of Focus Ultrasound around the world, and I'll show you more of that. So Focus Ultrasound is really paradigmatic of what we're trying to do, and that is transition from the old way of doing things in medicine to new and future way of doing things. And what does the old model look like? Multiple days in the hospital, invasive operations, in the case of brain cancer, toxic chemotherapies, trial and error treatments, where we don't really know what dose and modality we're doing, but we're doing it to try to help patients, and treating symptoms rather than diseases. So really symptomatic approaches. Focus Ultrasound can address each of these. It can convert them into same-day procedures, non-invasively, lowering the doses of chemotherapies in a personalized way, and gives us the potential, as you've heard from Dr. Obara, to actually modify the natural history of these diseases rather than to just treat them symptomatically. So this is the device that we're using currently at Sunnybrook, and it's the most commonly used device. And Dr. Heinonen is a pioneer and helped invent this device. It's a phased array helmet. And two things I'll point out. It's all done using real-time MR guidance with 3 Tesla MR. And we use over 1,000 individual transducers. Okay, so that it's not trivial. It's 1,000 individual sources of ultrasound that we can individually steer through the skull into the brain. One of the first examples that showed us that in fact we're onto something and that we can potentially change the way a disease is treated is essential tremor. So essential tremor is a common disease affecting the upper extremities and is a debilitating and disabling condition where people are really left entirely incapacitated by their tremor. Treatment majority of patients, even though that works, surgery becomes an option. It becomes an option in patients that don't tolerate or medications don't really work. So focused ultrasound, what that allows us to do is non-invasively and directly ablate and kill the part of the brain in a very focused way, driving that tremor without an operation. And this is that very same patient a few minutes after their procedure is done. So essentially cured of their tremor. And this is a durable, long-lasting, life-changing effect made possible by this less invasive approach. So just to give you a sense of how disruptive this has been in our field, we asked Sunnybrook Research Institute for the permission to treat the first patient in Canada in 2011. We treated the first patient a few months later. We published our first report of the first series in Lancet in 2013. We took part in a major trial, multi-center trial with several of our colleagues and helped lead this trial, published ultimately in New England, and got regulatory approval for this in 2016. So that 5 or 6 year period from initial idea and application to regulatory approval is about 1/3 of the time it takes a typical device or drug to come to market. Really a reflection of the tremendous impact and potential and disruptive potential of the technology. So then we got the idea, can we use this technology in other diseases and other conditions where making a lesion in the brain may be effective? And the answer, of course, is yes. So we know that for depression and OCD, for example, in patients who are treatment refractory, for whom medications and psychotherapies and conventional treatments don't work, surgery becomes an option and has been an option for many decades. But it's always been an invasive procedure, an open brain surgery. Now we can achieve the same objectives without an operation by lesioning those very parts of the brain that we know drive mood and anxiety. And now we've shown that it's possible to do that in a personalized, image-guided, safe and effective way to directly intervene in and influence those circuits driving mood and anxiety. And we're continuing to treat these patients on a regular basis and now have the world's largest experience, in fact, showing that it's safe and effective way to treat patients with refractory mood disorders. So we can non-invasively target discrete parts of the brain with no skin incisions in a personalized way as an outpatient procedure, really bringing to fruition the work that you saw Dr. Heinonen talk about that was shown in the lab, shown from a technical perspective, but now we've proven that we can do it in humans. The next question is, can we capitalize on the tremendous work that's been done in animal models and in preclinical models to use the same technology to deliver much needed therapy to the brain? So we've heard a lot already in the last hour or so about the blood-brain barrier. Needless to say, the brain takes up about 1/4 of the cardiac output with every heartbeat. So a lot of blood vessels, okay? What that is a lot of challenge for potential therapeutics to get into the brain, particularly where you need them the most. So we've seen already that focused ultrasound in preclinical models can give you that window, can open a temporary window using injected microbubbles into the brain, allowing you to potentially deliver therapeutics. And one of the things that we asked, well, can we use this technology to help deliver therapeutics in cancer specifically? Why cancer? Not only is it a major burden, a public health burden, not only is it deadly, but it's common. But we also know that some chemotherapeutic agents, one of them is Herceptin, which is an antibody, very effective at controlling cancer throughout the body, except the brain. In fact, most patients with breast cancer, metastatic breast cancer, they die of their brain disease, not from their systemic body disease, because chemotherapy agents can't get into the brain. So we conducted a phase 1 trial, first in human, global first trial, to show that we can in fact open the blood-brain barrier in patients with breast cancer and get drugs into the brain. So this is what that looks like as an example. This is one of our patients with a brain tumor and a highly eloquent, difficult to reach part of the brain. And this is, here we are opening the blood-brain barrier, and you can see this color map showing that we can, in a very focal, precise, way, open the blood-brain barrier just in the tumor, preserving areas around it. You can see here that the blood-brain barrier is open because gadolinium, which is a contrast agent, is able to seep into this area effectively. And you can see this white fluffy cloud stuff around the tumor indicating that the blood-brain barrier is open. And 24 hours later, it reverts back to baseline, indicating that the BBB is closed. So safe, temporary opening of the blood-brain barrier. Now, the neat part of this study is we were actually able to tag Herceptin, tag the chemotherapy with a radioactive ligand that we can visualize. So that for the first time, we can see the chemotherapy in the brain in real time in a living patient. And here you can see no chemotherapy, no Herceptin getting into the tumor. It pools in areas that are highly vascular in the veins and in the nasal epithelium, but not. Immediately after focused ultrasound, you see it significantly light up. Okay, so when we measure this, some areas see a 450% increase in concentration of chemotherapy in that part of the brain. So it's really the first human evidence, direct evidence of a passage of a large molecule antibody across the blood-brain barrier in cancer. So in cancer, so all bets are off. We are really tackling this across the board. We're interested in primary brain tumors, secondary brain tumors, and pediatric brain tumors as well. One of the most promising aspects of focused ultrasound in cancer happens not only to be the therapeutic potential of it, but also the diagnostic potential of it. And that's something we're also interested in, using ultrasound as a kind of biopsy of the brain. We know that not only does the blood-brain barrier represent a challenge to get things in, it's also a major challenge to get things out. And there may be the possibility of detecting peripheral biomarkers in our bloodstream that may give us a sense of what the diagnosis of something is in the brain. Why does that matter? Well, it matters to tumors like this that are very deep in the brain. And we need to know what this is because the pathology is either a malignant brain tumor that's inoperable or something that's very, very treatable with chemotherapy. To know that, we need a piece of it, but that requires a very risky operation. So can we use ultrasound to open the blood-brain barrier and detect pieces of that tumor in the bloodstream? And there's very early evidence to suggest that yes, in fact, we can do that. We can detect significantly more amounts of what's called cell-free DNA in the bloodstream after opening the blood-brain barrier. Cell-free DNA are fragments of DNA, potentially fragments of cells that we can then analyze and make the diagnosis without opening the head. So very first evidence that we can use ultrasound to make that blood-based diagnosis. Here's one of our patients with very early signs that we may be able to have an impact on tumor size. And it got us thinking that we can potentially use this technology for other brain disorders where the BBB is a major challenge. One I'll briefly mention is Parkinson's disease. We know, for example, that enzyme replacement therapy is a promising therapeutic for Parkinson's disease. It may be that some enzymes are missing in the brains of patients with PD, and just getting them into the brain may help offset or prevent some of the pathophysiology, some of the pathology of the disease. But GCase, or glucocerebrosidase, is a very large compound, cannot get into the brain in sufficient concentrations. So we conducted our first trial of opening the BBB in a structure called the putamen. Here we are opening the BBB. And although we can't tag GCase, the enzyme, to see it like we did with Herceptin, we can indirectly measure its impact by looking at how the brain uses glucose with a PET scan. And we know that as Parkinson's disease progresses, some regions of the brain become much more active. They're hyperactive, indicated by this dark red maroon area here. A month after the treatment in the area that we treated, that activity has been significantly reduced, indicating that we are having likely some kind of biologic effect. Interestingly, of the 4 patients that we treated in our pilot trial, 3 of them saw significant improvements in their motor scores by 6 months. And the one that saw the greatest effect was the one that was most deficient in their enzyme because they carried a genetic mutation missing that enzyme. So very early promising data. This is truly collaborative work done with our partners at University Health Network and got picked up by media. Very fortunate to be a part of this collaboration, which is ongoing. And we currently have a Western Brain Institute funded phase 2 trial looking at this question. So changing the treatment of brain disease is our goal, and global leadership and impact is the result. I'll conclude by just a few words about what we mean by impact and why we're here today. So we can define impact in different ways, of course. One is venues like this and public engagement, and we're very fortunate to have piqued the interest of the public every time we publish or do our work. The second is scientific impact. And again, we're very fortunate that our colleagues in our peers in the world of focused ultrasound and clinical neuroscience have agreed that this is a promising technology. Another way is looking at publications in the literature. And when we recently looked at the number of publications per year in focused ultrasound in the brain, we see this exponential curve over the last 30 years. And when you superimpose the major achievements in the field, both preclinical and clinical, you can see that the ones in red are driven by Sunnybrook and Sunnybrook researchers. So we're really proud to say that we're very much at the epicenter, very much at the global hub of work happening in this area. This is the global landscape of focused ultrasound currently. These are the most active centers around the world doing focused ultrasound clinical trials. Here we are in Toronto in red, showing the most number of dots. Each dot is a unique indication where a clinical trial is moving forward. And we're trying to really expand in breadth. Centers that have visited Toronto to learn about focused ultrasound, to take our protocols, import them back to their centers and start their own programs. So very, very much again, ambassadors of this technology. Now, something we're quite interested in is developing a national network. So we have ongoing partnerships with centers throughout Canada, very much interested in developing a common language, common protocols, common studies across the country to really advance this, make this much more than a Toronto, much more than an Ontario story, but truly a Canadian and made in Canada story. So are we done? So are we established that, the focused ultrasound can have an impact? I think, safe to say, not even close, even though we're very much interested in neurodegenerative disease, movement disorders, cancer. We have a strong interest in seeing this technology applied to stroke, to traumatic brain injury, and other indications where the BBB is going to be a major challenge for the delivery of therapeutics. So much more to come in the time ahead. I'll conclude by thanking the tremendous team that we have here at Sunnybrook that make this work possible. It's an absolute privilege to work alongside Professor Obara and Professor Heinonen as well, and the clinical leaders that really lead this area in all of the domains that you saw today. So what does the future look like? Does the future look like this? All of us with individual helmets, as Dr. Heinonen said, that we can nightly get a tune-up of our brain. Maybe that's far-fetched for now. Maybe it will happen in the future. But it's our view that personalized, image-guided, safe, and effective ways of treating the brain, of directly accessing this brain pathology is a very realistic goal in the near future. And we hope that by the end of today's session, you'll share that hope with us. Thanks very much.
Okay, thank you very much, Dr. Heinonen, Dr. Obara, and Dr. Lipsman. That was a fantastic set of seminars. And we're going to continue on now with a question and answer period. And we're very privileged to have two trainees join us here today who are going to moderate this session. So I'm just going to start by introducing the two trainees. And just a reminder that you can also add questions into the chat in the Zoom window, and we'll try to get to as many of them as possible. So the moderators will read the questions, and then we'll have Dr. Lipsman, Dr. Obara, or Dr. Heinonen come to the podium. So please indicate who you would like to address your question to. So I'm going to first introduce Dr. Ricky Kofood, who is currently a postdoctoral fellow in the laboratory of Dr. Isabelle Obara, and I probably butchered her name, so I apologize. I'll have her say it herself when she gets to the podium. So Dr. Kofood completed her PhD at Aarhus University in Denmark, where she also completed a master's degree in molecular medicine. Her research in Dr. Obara's lab is focused on gene delivery in animal models of neurological disorders, and she uses focused ultrasound, which is the subject of today's lecture. So she also studies how this method of therapeutic delivery to the brain can be used to administer gene therapy vectors. And her postdoctoral work was a natural extension of her previous investigation of therapeutic strategies for Parkinson's disease during her PhD project. So we thought we'd have a few fun facts about our two trainees. So Ricky's fun fact is that she eats at least 2 apples a day, so she's very healthy. And our second trainee is Dr. Yassine Touhari, who is a research associate in my laboratory. So he completed his PhD in the Centre de Biologie de Développement at the University of Toulouse in France, where he also completed an MSc equivalent. His research is focused on understanding how the retina develops and also on how these developmental principles can be used to design new therapeutic strategies for the disease retina. And his prior work focused on the developing spinal cord, where he first became intrigued about how the nervous system develops. And he's developed now an interest in gene therapy that has led him to also study how focused ultrasound can be used to deliver gene therapy vectors to the eye. So I'm going to have, and I have to announce Yassine's fun fact, which is that he's a very serious researcher, except when he's playing soccer and ends up being injured, which happens more often than we would like. So Yassine and Ricky, please come to the podium.
Thanks, Carl. So the first questions will be for Dr. Obara. And the question is, in simple words, how is Alzheimer's disease triggered? And why do some people get it and some people not?
Thank you, Yassine, for your question. So we don't know how Alzheimer's disease is triggered. We know that genetically there's less than 1% of the Alzheimer cases that are due to some mutation. So we know that. We know that some of the protein that we talked about, amyloid and tau, can be, that are pathologic, they can be toxic, and they are contributing to the disease. In terms of how to prevent some of the risk factor, aging, we cannot prevent this one. But there's other things that you can do for your lifestyle, like eat well, sleep well, exercise. All of these greatly contribute also to healthy aging. Oh, one more. Okay.
While you're here. So there was a question in the chat. Specifically, they asked how long time after the ultrasound treatment did you see the effects on neurogenesis? And I think I want to expand that question a little bit, and maybe you could talk about how long lasting these effects on neurogenesis are.
Sounds good. I'm trying to think of my timelines. So the first one is we've seen this effect on neurogenesis after 28 days was the earliest time point that we looked at. So ultrasound treatment was done at day 1, and then we saw neurogenesis at 28 days. And then on the multiple treatments time, that will be longer than that. I think it will be about 2 months, but I will have to go back to the publications to see the real timeline. But I would say I'm expecting that this neurogenesis is triggered between like 3 to 4 weeks, and it could last up to 2 months. What we found also with the therapeutic is that ultrasound is actually having an impact mainly on the cell proliferation for the neurogenesis, and some of the therapeutic are actually helping the survival. So we start to dissociate this mechanism as well.
Why still here?
Look at me.
What do you see as the greatest advantage? I think you just talked about it, about proliferation, but what is the greatest disadvantage of this disease, or something that we should be monitoring if we want to bring it to treat these diseases?
You're asking advantage and disadvantage?
Yes.
Yes. Well, okay. And maybe Clairvoyant can add on that as well. But, obviously one of the main advantages, it is non-invasive, and it is so adaptable to the different pathology, different brain areas, so without having to do any surgeries. So I think that's a main advantage. The main disadvantage, and it comes back to what Clairvo was saying as well, is like, how do you control to make sure that there is no damage? But I would say now with all the control mechanisms that are in place, we're getting to the refinement of this technology that is quite safe. But of course, you can use it from all the way to being inert and not damaging the brain if you don't have the right parameters or if you don't have the right control to monitor it, it could be damaging. So care should be taken dealing with the brain for sure. Yeah.
Okay, then I think we would like to have some questions for Colero. So the first one is, practically speaking, what does it require to have this treatment at a hospital?
You mean on long run? So it requires that you have a system and you have a neurosurgeon or somebody who is willing to use it clinically, and then a technical team to help, and clinical team like neurosurgeon. So it requires a significant team, but that question is probably more better for you.
Yeah, also, Doctor, you're just talking about a team. So we saw from your presentation that it involves clinician, physicists, and like a lot of people. How practically the interaction between these people, because they speak different language, right? So how practically they can put together in a room and collaborate?
You just put them together in the same room, and there is a patient. So I guess Nir, you could explain your experience, but it seems to me that the first treatment, the first case, it's kind of you learn your way, but very quickly people are starting to work together. Yeah, and the team works really nicely these days. So.
Okay. And we do have a question from the chat. Very technical, actually. So what are the microbubbles made of?
So microbubbles, they are gas bubbles. So these are perfluorocarbon gas. They could be air bubbles also, and they are encased in a lipid cell. In this case, they could be also albumin cell or other materials. So these are pre-made microbubbles of 2 to 3 micrometers that are biocompatible, can be injected in bloodstream. They break, they go a couple of cycles through the circulation and break slowly, and the gas is exhaled through the lungs, and the cell material is like normal lipids, so they are disposed by the body.
Then we have some questions for Nir. So if you are a patient who are receiving the focused ultrasound treatment, what can you expect from a treatment day? Like, can you go home the same day, and will you have to be anesthetized?
Thanks for that question. So we're talking mostly about the ablative sort of the indications that are furthest along. We don't use a general anesthetic. We use a local anesthetic and conscious sedation if we need to sedate patients to make them more comfortable. But patients are awake during the procedures. We need them awake because we communicate with them. So we need to make sure that there are no untoward effects, but also we need to monitor for clinical effects. So that's a major advantage of this. So you don't need an anesthetic. You don't really need a recovery room. Patients come to the hospital. They're treated like they're going to go to surgery. They come to the focused ultrasound suite. They meet the team. And then they undergo the procedure, and then usually they go home within a couple hours of the procedure. They're observed. No, we still treat this like a neurosurgery, like an actual operation, even though we don't open the head, because we've done hundreds of these, but we haven't done thousands of these. So we want to make sure that patients are monitored carefully, that they're carefully observed. But so far, things have gone pretty smoothly, and patients tolerate the procedures quite well.
Yeah, we have questions from the chat, actually. Right. So for treating brain tumors with fuzz, to what extent does opening the blood barrier solve the problem of getting sufficient drug to the tumor? In other types of solid tumors, there are large gaps between the endothelial cells, and yet there is insufficient drug uptake. So also to what extent does success in treatment depends on the concentration of the drug in the microvessels?
All excellent questions. There's certainly blood-brain barrier disruption in the context of brain tumors, but it's in a very disorganized fashion. You have breakdown of blood vessels, et cetera. We know that all of the patients that we've treated in our trials are post-operative, which means that they've had their tumor resected and there's a rim of tumor around, and sometimes there's a recurrence or there's some nodularity around there. We know that 98% of the time, in the case of malignant primary brain tumors, recurrences occur within 1 to 2 centimeters of that rim. Okay, so even though there may not be enhancement there, we know that there are tumor cells there. So that's what we target with focused ultrasound, the area of the rim that we've resected, plus a little bit beyond, knowing that there are tumor cells, and that's where most recurrences will occur. We want to open the blood-brain barrier in a way that's very controlled, in a way that's very systematic, and hence the very tight control of the microbubble dosage and focus parameters that we try to optimize. A lot depends on the dose for sure. We have the preclinical data to suggest that we see several fold increases of chemotherapy, but until we can actually measure, we don't know that for sure. So there are many levers we can press with this technology: microbubble dosage, chemotherapy dosage, technical parameters in terms of cavitation doses and things like that. All of that is very early days, but a lot of potential work to go into this to optimize the procedure.
We have one more question from the chat that I think is very relevant. Are there any risks or added barriers for individuals with atherosclerosis?
Also a very good question. So presumably cerebral atherosclerosis is what we're talking about here. We think that anything that would increase the risk of bleeding may represent a material risk. So we have an ongoing trial of opening the BBB in patients with Alzheimer's disease, specifically targeting areas that are amyloid positive on amyloid PET scans. So amyloid renders blood vessels very fragile. The leading cause of intracerebral hemorrhage is cerebral amyloid angiopathy. And now we've seen that even when we open the BBB in areas that are amyloid positive and amyloid rich, we can open the BBB safely, reversibly, repeatedly over multiple times, and it has not been associated with a material increased risk or any risk of bleeding, swelling, or other concerns such as ischemia in that area. Now, in those studies, we specifically exclude people who have so-called microbleeds. If we do a scan and we see that there's a lot of microvascular disease, as indicated by microbleeds, we exclude them from participating precisely for the reason that you mentioned.
On the same area, when you're treating Alzheimer's disease, do you see any trends in success between men and women?
The numbers are too small right now for that. Our studies are designed primarily to study safety, reversibility, technical feasibility. It's an important question and one we're interested in. We're conducting a trial now where we're just opening the BBB. The next phase is going to be to deliver a therapeutic, again, leveraging the very compelling work you heard from Dr. Obara in terms of IVIG. But certainly gender differences or sex differences are ones that we're very interested in studying.
Yep. So now we would like to ask some more questions for Isabel from the chat. So there is a lot of questions on different diseases, but one disease that I see a lot of people being interested in hearing more about is focused ultrasound for stroke. Could you talk a little bit about what you think about the potential for that?
Focused ultrasound for stroke, yes. I think it could be used especially for, when stroke cannot be treated acutely and stroke get into a phase of rehabilitation that is stagnant sometime in terms of regeneration. So I think we could think of it in a way that once the stroke is over, the blood-brain barrier is resealed, and how can you go in with focused ultrasound and modulate the environment to re-trigger this regenerative phase that we can see happening with both the glial cells and the neurons, and even give therapeutic to that zone and make it more plastic, more malleable, and more conducive to regeneration. So that's one way that I would see stroke. Clairvaux has done some study in stroke, even with focused ultrasound, that are completely different. I think, Clairvaux, you should talk about that too. Because it's different. I think it's interesting.
Yeah, so one option is in acute stroke, if you have blood vessels closed, is to use focused ultrasound and open the vessel. So destroy that blood clot as is there non-invasively. That is, people have shown the feasibility of that in preclinical models, but it still is technically very, very challenging with the current devices. But the technology is getting there, so maybe one day we can have focused ultrasound systems in ambulance, and they could be used to just zap the vessels and open the start the circulation. Who knows? We are not there yet. So.
Yeah, so we have a question. You are in close contact with the patient. So what are their questions, their concerns, or their fears when they come for the clinic?
So I like this question a lot, actually, because as you said, in alluding to the earlier question you asked Dr. Heinen about, the team, speaking different languages, et cetera. One thing that I'll just say that it's true. I mean, we work with physicists and technicians and different clinicians from different areas. But we're all focused on the same thing, which is the patient. And it's a nice symphony that happens, when somebody comes the morning of the procedure, seeing everything come together and gather around the patient, focus specifically on treating them, whether it's an experimental treatment in the case of drug delivery or Alzheimer's or an established treatment like we do for tremor. So I can tell you that, for the patients with tremor, they want a safe procedure. Most of their questions, everybody's questions actually are focused on safety. Is this safe for me? Is this going to work for me? Those are the two questions people ask. We have a lot of data in tremor, that yes, it's highly effective and it's looking like it's safe, certainly safer than the traditional approaches, which is an open neurosurgery approach. Pretty confident in that area. For the drug delivery, people want to know, is this right for me? Is this safe? What is your experience with this? It's a conversation. It's a dialogue we have individually personalized with every patient. And we have an extremely fruitful and collaborative relationship with our research ethics board, with Health Canada. All of these studies are Health Canada and RAB approved, and really go through all those steps. But all patients are focused on, is this going to work for me? Is this safe for me? And it's a constant dialogue with them.
While you're here, we have another question specifically for you in the chat. So this person is asking, how are the effects of the focused ultrasound ablation different from the pallidotomy, and are there any side effects of chemotherapy in the brain?
Okay, somebody with very good knowledge of stereotactic surgery. So pallidotomy is a lesion of the pallidum, of the globus pallidus, and a highly effective treatment for patients with Parkinson's disease who have dyskinesias and who have specific motor symptoms. What we lesion in tremor is a thalamotomy. We lesion the VIM nucleus of the thalamus. So it's a different anatomic target in the brain. So VIM is lesioned in patients with a central tremor or with tremor-dominant Parkinson's disease. Pallidotomy is done in patients with Parkinson's disease who have very specific kinds of troublesome symptoms. It wouldn't be done in patients with tremor. The tremor effects are going to be similar in patients with Parkinson's disease. So there's excellent tremor control with pallidotomies. And the second question was about chemotherapy. What was the question?
Specifically, the side effects.
Side effects. Yeah, so very important question. For glioblastoma primary brain tumors, we use an oral chemotherapy agent, very, very safe, very good track record, and we have not found any untoward effects. We are very interested in delivering agents such as doxorubicin and other chemotherapy agents that do not get into the brain at all. And in those cases, we certainly are interested in what effects would have on the brain, in the tumor and beyond the tumor, and there are different mitigation strategies we can use in terms of the volumes of BBB that we open and the doses that we use, et cetera. That's really the purpose of the phase 0, phase 1 trial, to answer those very questions, which we're quite interested in.
Thank you very much. And there is still a lot of interesting questions in the chat that we were not able to address today, but I would like to direct you all to sunnybrook.ca/focused ultrasound, where you can read more about the work at Sunnybrook and also some of the clinical trials. So I would like to thank our speakers, Carol and Yassine, and the Canadian Association for Neuroscience, Sunnybrook, and the funders, and the patients that make this work possible. And I would like to thank all of you who joined us online today. And I hope you learned something and are as excited about all the possibilities of focused ultrasound as we are. And I hope you have a great evening. Thank you.